Hi everyone, welcome to the live lesson. I knew I had to squeeze in one of these last-minute.com sessions for everyone out there. Thank you for joining the live. I'm going to try and do as much as possible in this live because I know a lot of you have got so many different questions and you've got work to do tonight. So I'm not going to make this a very, very long live. I'm going to try and be as quick and precise as possible and get to everyone's questions as quickly as possible. Remember, there is a chat alongside that you can ask all the questions that you want. I'm going to try and answer them as many as possible. If you know people that want to join the live or if you've been waiting for me to go live, then share the link with them now. Make sure that your quality is set to the highest it possibly can, just because it means you'll be able to read what I'm writing and read the questions that we'll go over together. A lot of the questions I'm going to use today are some of the ones that I included in my prediction video. So if you haven't gone and watched the prediction video, I suggest you go and do that after this. If you can't stay for the full length of this video, that's okay. When we're done, it will be uploaded automatically, and then you can come back and watch it as many times as you need. And you want to hear me explain things. And of course, I have the full library of all my other YouTube videos as well. So don't stress everybody. Okay. Whilst everyone is joining the live and a couple more people will, I'm just going to tell you some of the things that we're going to cover today because I'm pretty sure you're keen to just hear some of the general items. I'm going to cover things like the eye, specifically accommodation. I'm going to do the semicircular canals, so that's for balance. I'm going to cover geotropism. I'm going to cover a female menstrual cycle graph question, and then I'm going to do like a reflex spinal nervous system question as well for you guys. Yeah, that's basically what we're going to cover. That means we've covered the big 4 things. If we have enough time, I might even squeeze in a little bit of endocrine. Just as a top tip from me to you, is endocrine is often understudied, everyone. So I suggest that you go home and you're already home probably, and make sure that you are covering it as much as possible. Okay, so without further ado, because I don't want to waste any more time, I'm going to share my screen with you. And we can get into it. Right. Okay, I think you can all see my screen. Okay, so this is our live lesson, everybody. Just hold on a little OROS for energy. Okay, right. So without further ado, I am going to dive into our first question. And I want to start off with a section that I think many of us don't study enough, which is going to be the geotropic section. I think plant hormones don't get enough attention, and I think we're going to underestimate it a little bit tomorrow. Okay, so let's dive in. So this is a past paper question. I took this particular question from the Eastern Cape, September in 2023. It says an investigation was conducted, and this bit right here is important to us, to determine the influence of external stimulus on the direction of growth in plants. Everybody, that's the aim. So in your exam tomorrow, always highlight that part of the sentence. We had the following procedure. We had germinating seeds that were pinned onto wet tissue, as shown in the diagram. The disc was placed vertically in an upright position in a dark room. Seedlings were gently sprayed with water daily. After 4 days, the discs were turned in a position shown in diagram 2. After a week, observations were made. Direction of growth of the young stem and root were recorded. So let's move this up so we can see the rest of this question. Okay, so this is, this is before, basically, as we start at the beginning, and this is after, in other words, 4 days after they have moved it around. And so essentially, diagram 1 is showing us the initial, and then diagram 2 is showing us how they've turned the disc clockwise and kept it stationary. In other words, they are turning it and stopping and turning it and stopping. It's not continuously revolving, which is an important thing to remember. Okay, so let's go into the geotropic effects of this. Now, I kind of already know it's going to be geotropism because they've removed the light, so there's no light affecting the outcome of this. Okay, so it says state the pattern of distribution of the growth hormone at A and B. So first of all, remember the growth hormone they are talking about is the auxins, right? And now what you need to be able to do is tell the difference between the distribution or the pattern of distribution of the growth hormone at A and B, meaning there's a difference between auxins in a shoot and different distribution of auxins in a root. Okay, so when they say state the pattern of distribution, what they mean is where is it collecting and where's it distributing to. So if A is the shoot, that means it's going to be in the apical bud, and that means it is going to be moving downwards, right? Because you make it at the top. Then if you look at B, it is made at the apical root. I suppose you could also call it an apical bud as well. And that is also moving downwards. And what I mean by moving downwards is that it's diffusing from the tip down. So another way to write this as well, I'm just going to write it on the side here, is that when we talk about shoots and auxins, auxins we're going to move towards gravity, basically, for both of these. Okay. Essentially, they also want to know is it equal, is it even, and it's only one word, it's only one mark, everybody. So don't overthink this. They're not trying to fool you. They're not trying to ask you very complicated questions here. They just want to know what is the pattern of distribution of the growth hormone. In other words, it's starting at the top and it's evenly distributing down lower. In the root, it's starting at the bottom and it's not distributing up, essentially. Okay, it's not going up. That's the point, because auxins don't move away from gravity, they move towards it. So don't feel like it's wrong if you repeat yourself twice, is what I'm trying to get at. That's what they're trying to confuse you with. And yes, they will try and confuse you. Okay, next question. Explain one reason why the disc was kept in the dark room. You may remember that from earlier. And essentially, everybody, remember when we use an explain question tomorrow, please remember that you must give a statement and then you must follow it with a reason. That's for 2 marks. If it's out of 3 marks, then you must give 2 reasons, 1 statement and 2 reasons. So why was the disc kept in the dark room? So, hold on, let's get rid of that little box. There we go. The statement and reason that we're going to put there is we are going to say to remove the light stimulus. And then the second tick after that would be so that only, I've got to squeeze this in, gravity will affect the growth. It's as simple as that. We put it in a dark room so that no phototropism is occurring, only geotropism is occurring. Okay. Then it says for number 3, draw a neat diagram to show the direction of growth of the young shoot and root in diagram 2 after a week. So I'm not going to redraw the whole thing, but I'm going to go a little bit closer so you can see it. If this is the distribution of the auxins, and we've got to pay close attention, this side here is the root, this side here is the shoot. Okay, you got to pay close attention to which side is which. If they have moved it and now they're saying they're going to leave it in this position for a week, what you should then draw is the root going down and the shoot coming up. That's basically the sketch that you would have made. You would have just redrawn the bean and then, or the seed, and then you would have had the root and shoot coming out, making sure that you orientated yourself with which one is which. If you're asking yourself, ma'am, why did they even rotate it in the first place? It's to see whether or not you are spatially aware, which means, did you make a note of the fact that the bean was in this position first? And then did you notice it revolved and is now in this position? It just makes sure that you're paying attention to where the root is before and after and the shoot is. It's as simple as that, everyone. Okay, so that's all you would need to do. Now, just to make sure that you get 3 marks, please always assume that a diagram must have a heading. It must be drawn in pencil. And I strongly suggest including a label. In actual fact, even though this is out of 3 marks, I would label it with at least 2 labels and a heading, and I would label the shoot and the root. I would. Or I would use the letters A and B, because they also use the letters A and B up in the top part of the diagram. So I would label which one is A and which one is B. Okay, right. And then last but not least, the big one, right? They always like to end a question off with a very chunky 7 to 6 mark explanation. And now what you are going to do is you are going to be perfect in this answer tomorrow. If you do this, you will always get your full marks. It says, explain the direction of growth of the young root, okay, after it was placed in the position shown in diagram 2. So remember, we're only talking about diagram 2, so we must refer to the fact that in diagram 2, after it was placed in the position shown in so in other words, after time has happened, what's going to happen? You are going to explain why the root over here is actually going to turn and bend downwards. All right. So how do we get full marks for this? So let me give you some bullet points that you would include. So number 1, you need to tell me, where are the auxins? So the auxins are going to move towards gravity on the lower side of the root. Okay? Because remember, they're attracted to gravity, and you can include that, and you can say, as auxins are positively geotropic, which means they move. No. Yes. Yes, they are. They move to, sorry, I'm just clarifying in my mind. They move towards gravity. Okay, so auxins move towards gravity on the lower side of the root, as auxins are positively geotropic. Next point. These cells on the lower side with the high, put your concentration of auxins do not elongate. That's important. So they do not elongate, because remember, they're not going to get long. And if you can't remember what that means, remember, cell elongation is when a cell goes from this to this. Okay, so they're not going to do that. It's the ones on the lower side. And then cells on the upper side will elongate, causing the root to bend towards, sorry, I'm writing this very quickly, gravity. Okay, now a good rule of thumb to get all your marks is to tell me, where are the auxins in there? So where are they? Okay, they're moving towards gravity. They're moving to the lower side, which has the higher concentration. So it's always tell me where they're moving to. Tell me where that is. That's the higher concentration. Tell me what's happening on the opposite side. Once you've told me that there is a high concentration on this side and a low concentration on that side, tell me what's happening with the cells. One side, the cells are elongating. The other side is not elongating. Because the one side elongates, it causes the root to bend. And in this case, it's bending towards gravity. If it was light, of course, it would be bending towards the light. And so you've got to really, 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 really know your geotropic explanations, everybody. And there's no reason why you wouldn't get 7 out of 7 for this. Okay. So that is geotropism in a nutshell. There are many different ways they could ask you this tomorrow. They haven't asked geotropism in a while, so it makes me feel like they might ask it. They've been loving the questions also a lot lately when they like cut the tip off as well. That's another one that I've seen a lot of. So I think, my screen is frozen. I think that if you guys want to prepare as much as possible for your exam for tomorrow, I think you need to also be prepared what happens when you cut the tip off of the shoot or the root. Okay. So I'm just going to pause for 2 seconds and ask you, sorry, everybody, I've closed my page. I will come back into it. Don't stress, everybody. I'm just going to stop sharing my screen for a minute. If there's any questions you want to ask me about geotropism, please do that now while I just refresh my page so that we can go back in again and that we can have a lesson together. And I can see the chat, so I can, of course, see what you want to know about geotropism. Oh, quick response to this one question I can see here. Yes, this video will be uploaded when I'm done. So if you want to come back and watch it, you don't have to watch it all now. You can watch it later. So Geo, what happens when you cut the tip off of a shoot or a root is you remove the auxins, which means there will be no geotropism, there will be no phototropism, there will be no bending, basically. And when you also cut the tip off, it stimulates lateral growth, which means it stimulates side branches or lateral roots. Geotropism. So this question says the geotropism is only to do with gravity. Yes. But you do need to know that shoots are negatively geotropic, positively phototropic, which means they grow towards the light, away from the ground, and then roots are the opposite. Roots are negatively phototropic, positively geo, which means they grow towards the ground, away from light. And that's what keeps plants above ground and their roots below ground. Okay. For some of you who wanted to know where this is from, this question is from the Eastern Cape prelim paper or the September paper back in 2023. Okay, apical dominance, everybody. I can recap that very quickly for all of you. I just want to see if I have joined back to my live. Let's just see here if I've connected again. Okay, yes, I have. Let me share my screen again with you. Okay, so apical dominance is very simple. It is what allows trees to have this appearance. And so essentially that's because the apical bud is at the top here, and that's where auxins are made in their highest concentration. So you just have a really high concentration at the top of a tree, and as it diffuses down, it gets further and further away, which means it gets a lower amount. And because there's less and less auxins, it allows for lateral branches to grow out. So apical dominance, in a nutshell, stops lateral branches or side branches from growing. Why? If they ask you, why is apical dominance important to plants? It's important to plants because of photosynthesis. You don't want the top of your tree those leaves and branches, to compete for light with the ones at the bottom. Because if you imagine a tree being a square and not a triangle, it means that the sunlight can only get to the top part of the tree. It can't get into those deeper layers. But when it is a triangle, you're able to get a really nice, even distribution of light all the way over it. And that's what apical dominance is. All right, I think I'm going to leave it there, and I'm going to move on to our next section, which I know you're all waiting in bated breath here, is a female reproductive question. Now, I just want to also warn everybody that I feel personally that matrics spend too much time studying the female reproductive system. And when it actually comes down to it, the whole question is easy, except for one question, maybe out of 5 marks. And you've spent too many hours worrying about it and not worrying about the other topics. And I think that you kind of shoot yourself in the foot a little bit. So I am going to do this question because I do think there will be some kind of graph question tomorrow. And I think, as I suggested, it might be a FSH question, maybe even linking to estrogen and how they work together, or should I say how they're linked to one another. But yeah. Let's dive into this, because I know you're going to have a lot of female reproductive questions, and I want to get to them with enough time to move on to the others. So let's dive into this question. Unfortunately, I can't remember which paper I got this from, but this is a NSC final November paper that I did get this from. So it says the graph below shows the levels of 2 hormones that are secreted by the pituitary gland during the menstrual cycle. So they've already given you 2 clues as to what these are. So we know that there are pituitary hormones. And in terms of the female reproductive system, there are only 2, and that is FSH and LH. Now, on the graph, a lot of matrics struggle to tell the difference between which one is which, right? So I have a very, very simple method of telling the difference, and I'm going to start with the easier one of the 2 to identify here. When you are looking for LH peaks once and once only, and it peaks near or around day 14. So looking at this diagram, there's only one hormone that's peaking around day 14, and that's going to be hormone B over here. So that is definitely 100% LH. Now, the next hormone, which you would go, oh, obviously, ma'am, it's FSH, because that's the leftover one. But I just want you to know something that bothers me and that matrics don't get right. And that is that if this wasn't LH and FSH, let's say it was FSH and estrogen, I know a lot of you can't tell the difference between FSH and estrogen, because if I drew in estrogen, not even that. Actually, I'm going to draw it better. Let me draw it better. Okay, that would be estrogen. And so a lot of people can't tell the difference between which one is which, especially if it's FSH and estrogen. So how do you know which one is which? Well, FSH is always going to peak first. Do you see how there's a peak here? And the other line that I drew in red there, which would represent estrogen, peaks second. So the very first hormone that peaks, the very, very, very first hormone is always going to be FSH. It's never going to be any other hormone. Then if we move on to knowing why it's also FSH, is because it peaks again on ovulation day, because it helps with ovulation and the corpus luteum. It also helps with the negative feedback, so that we start making progesterone and we don't make any more eggs, because we don't want any more eggs. So how do I know if it's FSH? Because it peaks first and it peaks twice. It peaks first and it peaks on day 14. Estrogen will never peak on day 14. As you can see here, it peaks early and then it peaks after ovulation. Okay, so let's get into the questions that go along with this diagram. First things first, it says state 2 functions of hormone B. So you had to correctly identify what LH is. I don't think a lot of people know the second one as much as they do the first. But first of all, the two functions are ovulation, which I think most matrics know. But I think the second one is where people struggle. Just remember, the word is luteinize, luteinizing hormone. What other structure has the word luteal in? Well, the corpus luteum. So it's there for it's there for ovulation and the formation of the corpus. Ugh, I always spell luteum wrong. There we go. Luteum. Okay. So just keep that in your mind, guys, because a lot of people know the one and not the other. Okay. Next question, which I really liked because it's often a question that we see in these final exams. They speak about like what's stopping pregnancy. I said in my prediction video that maybe they might have something about the fallopian tubes being blocked. Another question that I think might come up, not directly linked to this question, but linked to reproduction, is about the amnion, the amniotic fluid. And so like they might even do something where like women don't make enough amniotic fluid. And then they're going to ask you questions like, well, if she doesn't have enough amniotic fluid, what's the problem? What's the problem with the baby? It's not going to be able to maintain its body temperature. It's not going to be a shock absorber, those kinds of things. So if we move into the next one, you can see it says, explain why a female who is struggling to get pregnant, A, may be given pills containing hormone A as a treatment. Now, remember, it's an explanation question. And I always say this, statement and then a reason. But now it's out of 3 marks. So you've got to give 2 reasons. So make a statement. What does that mean, ma'am? It means... say what hormone A does. Make that statement. So what does hormone A do? I'm going to just move this over so I can fit this in. Hormone A is going to stimulate a follicle, right? So she's going to take these pills so she can stimulate a follicle. Now, let's follow on the reasons with this. Now then, because that follicle is going to develop an ovum. And then, of course, that ovum will secrete. Oh, I can't fit in the word secrete there. Let me just move this over a bit. Oh, secrete estrogen. And what do we use estrogen for? Well, we use estrogen, remember, to grow back the lining of the uterus. Okay, so why is she given it? Well, you tell me what FSH does. So statement. FSH stimulates the release of a follicle or the development of a follicle. Next, reason. Well, we need that follicle to turn into an ovum. And that ovum is going to make estrogen so that the ovarian cycle, so that the uterine cycle can grow back the endometrium so that she can actually get pregnant. Because there's no point in making an egg if it has nowhere to go. It has to go to the endometrium, and you can only grow the endometrium back with the estrogen. Okay, let's move on. Now it says, explain why a female who's struggling to get pregnant for the next one will have her levels of hormone B constantly monitored. Now that means, everybody, that you need to understand that we're talking about LH. Now, if she wants to be pregnant, right, doesn't she need to know when she's ovulated? So why? Remember, this is explain. So as always, a statement and a reason. So why will her levels be monitored? So to establish if she has ovulated, as this, hormone peaks on ovulation day, and therefore, I can't fit this all in, sorry everybody, without writing all over the place, increasing the chances of fertilization. So statement, we want to establish that she's ovulated because it will be the peak on her ovulation day, tick. Therefore, increasing the chances of fertilization. Or you could say, therefore, increasing the chances of the egg cell meeting the sperm cell. Something like that. Okay, right. Then I'm just going to move this up now because I need as much room as possible. It says for number 3, explain how the levels of hormone A on days 0 to 5 will differ in a pregnant female. So if we go back up here, right? Remember, everyone, hormone A is FSH. If you are pregnant, are you going to make FSH? No. No, because you're already pregnant, right? You don't need to be pregnant twice. You don't want babies at 2 different stages. So how will the levels from day 0 to 5 differ? So how will they differ? So I'm going to answer it down here. I'm going to say they will not rise as a ovum has already been fertilized, so as to prevent 2 babies at different stages. That's what we want to say. Different stages. So, pregnant women don't make FSH. They're already pregnant. They don't want to make another follicle because if they make another follicle, they will be pregnant with potentially 2 babies at 2 different stages. One could be 4 months, the other one could be 4 weeks, and we don't want that. So that's how the levels of hormone A will differ. FSH will differ. Okay, coolio. Now, I just want to remove some of this. I'm going to move it to the side. Just get my eraser is so small. Let's make the eraser bigger. There we go. Okay. So I want to do this very last one because I think this is the one you all dread. Describe the secretion of ovarian hormones and their role on the menstrual cycle. And that's for 5 marks. So do you know your ovarian hormones? Your ovarian hormones, remember, are estrogen and progesterone, right? So now they're asking you to describe the secretion of those hormones and then their roles for 5 marks. So I'm just going to bullet point it to keep it short and sweet. This little window is irritating me. Let me just get rid of it. There we go. Okay, so number 1, the ovarian hormone. Let's start off with estrogen. So you must always tell me where estrogen comes from. So estrogen from the Graafian follicle, next point, will regrow and maintain the endometrium. Sorry, I couldn't think of the word. Okay. Progesterone from the, I'm just going to put here CL, short for corpus luteum, will make the endometrium vascular and glandular. Let me just move this up a little bit. Maintaining the endometrium. Okay, So describe the secretion. That means that you need to say where they came from, which we've done. So from the Graafian follicle, and then the role in the menstrual cycle. So the role in the menstrual cycle for estrogen is to regrow and maintain the endometrium. Again, progesterone comes from the corpus luteum, which makes the endometrium vascular and glandular, also maintaining the lining. So we've got about 5 marks. Some of them are double ticks. For example, I would say we would go 1, 2, 3, 4, and 5, something like that. That's probably where they would put the ticks on that one. So that is the ovarian hormone cycle. It's not so scary, guys. I'm just going to look at your questions quickly while I take a quick little break and have a look. And I'm also checking my laptop power. I'm just having a look. Everybody, for those of you asking me for a scope, you should go and watch my prediction video. And also the scope of the exam in terms of the actual mark allocations is in your exam guidelines. Everybody should have that, and everyone has access to that. I'm just reading some other questions. So Yash asked, doesn't the corpus luteum also release estrogen from day 14 to day 28 as well? It does, but it's very minimal. It's very minimal, but yes, it does. You're not wrong. It does. I'm just focusing on its progesterone. Can you write in point form? I wouldn't write in point form, but I would like fake it. So in other words, you can pretend to write in full sentences whilst still writing in point form. Does that make sense? Like you write a sentence, full stop, and then just start the next sentence on the next line. Why do I not suggest writing it like I did now? Because sometimes the memo gives a tick for like a connector word, almost like a conjunction or like a word that's joining the one idea to the other. And if you don't write in full sentences, you might miss that. Okay. What somebody asked here, who is this? What is the negative feedback between FSH and progesterone? So it's a very simple negative feedback. As estrogen increases, progesterone, beg your pardon. As progesterone increases, FSH decreases. But I just want you to know that progesterone doesn't cause FSH to go down. It's actually estrogen. FSH goes down as estrogen goes up. What keeps FSH down is progesterone, because remember, progesterone is the pregnancy hormone. So if you want to stay pregnant and you don't want to make another follicle, you'll have high progesterone and low FSH. But who pushed FSH down first? That would be estrogen. Estrogen would push FSH down, allowing progesterone to increase. And when progesterone is at its peak, along with some estrogen, it keeps FSH down so that if we do get pregnant, we stay pregnant. Okay. Somebody asked here, if I say thicker instead of saying vascular and glandular, am I correct? No, you're not. You do need to say vascular and glandular. They don't mean the same thing. Thickness does not indicate if it has lots of blood vessels. It just means that it could be the tissue could be thicker. It doesn't mean there's any blood vessels or glands inside of it. Somebody else asked here, is LH only released on the 14th day? Generally speaking, yes, on the 14th day. Some women it's on the 13th or the 16th, but you'll know it because it's the only one that peaks once somewhere in the middle. Somebody asked here, is it advisable to write as much as we know? Sometimes. If the question has like this one up here, I just want to bring your attention. You see this one up here where it says state 2 up there? If you write more than 2, we will only mark the first 2. If, however, this one we did, this long one we did now for 5 marks, this one at the end. If you write like everything you know, you run the risk of not convincing your marker that you know what you're talking about. And so they might not be very lenient. They might think that you're just putting everything down and then waiting for us to figure it out. This is the rule of thumb. This is a beautiful piece of advice I would give any matric writing tomorrow. When you write a long explanation question for your marker tomorrow, are you painting them a picture with your words? Or are you letting them guess or imply or infer, if you want to be really fancy, what you're actually saying? So I need you to be very literal tomorrow. I don't want you to think they know that you know. You must pretend they don't know anything, and you must tell them from beginning to end. For example, when we did these explanation questions, tell me what estrogen does, tell me where it's made, and tell me what it does and where is it secreted to. I need you to really paint a picture. I need to be able to imagine this. If you put everything down, I might think that you don't know what you're talking about and you're just waiting for me to find the right answer. Okay. Please also remember for tomorrow's exam that all of you have gone over spermatogenesis and oogenesis, and only write the exam guideline answer. Don't write what's in your textbook. Don't write what's in your teacher notes, unless, of course, it's my note, because I write my note based off of the guideline. So only the exam guideline for oogenesis and spermatogenesis. Okay. So I'm going to do one last question, which I think is actually a really good question. What causes the corpus luteum to degenerate if fertilization doesn't occur? Is it that LH is low and LH maintains the corpus luteum? Yes. So if you don't fall pregnant, there is nothing maintaining the corpus luteum. After fertilization happens, a chemical reaction occurs where the zygote sends a signal to continue producing LH. Now, it only does this for a short period of time, and it's the time in which you have the embryo attaching to the wall of the uterus, or should I say the endometrium, because another one that matrics don't know, which I want you to know now, as I'm going to say it out loud, is the placenta takes over the job of the corpus luteum. The corpus luteum disintegrates about month 3. So it's completely gone in month 3 of pregnancy. And after that, the placenta takes over and secretes large volumes of progesterone. So I just want you to know that. Okay. I have a question here from Jess saying difference between corpus callosum and corpus luteum. The corpus callosum is in the brain. Corpus luteum is in the female reproductive system. Okay. Let's move on, everybody. I'm just going to do a quick little highlight reel of the eye. And this one is a mixture, if I look carefully, between, it's not a mixture, I beg your pardon, is it? No. This is accommodation. I'm just going to give you the highlight reel. So what you're looking at here is accommodation. And can you see how the lens is getting thinner here and it's getting fatter here? And so this is far vision and this is close vision. And essentially what you need to be able to do tomorrow is explain accommodation and what's happening to the lens. Remember that you have these, I'm just going to put here sus ligaments. So they are the suspensory ligaments, and they are attached to these muscles over here, which are the ciliary muscles. Now, when we talk about accommodation, I want you to know that if one is contracting, the other is relaxing. What do you mean by that, ma'am? Well, if the ciliary muscle is relaxing, then the suspensory ligament is contracting, or the better word is pulling taut, like it's being pulled on. And so if one is relaxing, the other one is contracting. And so that's what's happening when we look at close vision. And that's so that we increase refraction. What is refraction? Bending light. Why must we bend light? Because the more light you bend, the clearer your vision, everybody. This far away one is just the opposite. You can see the suspensory ligaments are like slack. Look how loose they are here, which means that the ciliary muscle is contracting and the ligaments are relaxing. And so that's what makes the lens fatter. And less light is refracted, and that's because when you look far away, you don't need to see as much detail. They won't ask you to draw the structure of the eye. They may ask you to label it. They may also ask you a question. It's not in this picture, but they may also ask you about the pupillary mechanism, which, remember, you've got circular muscles and you've got radial muscles. And the circular muscles are the muscles that make the pupil smaller, and the radial ones are the ones that make it bigger. And so you need to know that if I'm going into bright light, the circular muscles make it smaller. If I'm going into dark light, the radial muscles open up the pupil to let more light in, and that is the pupillary mechanism. It's about 4 marks most of the time. I'm not really too stressed about the eye, everybody. I wouldn't be concerned if I was you, to be honest. I think the hardest question that matrics get is just because they don't know what lens you're supposed to put in front of the eye. So if you have myopia, which is what I have, means I can't see far away, I need a convex lens like this one, which is exactly what my glasses are right now. It's a convex lens, so it goes in. A lot of matrics don't know that. So please know how to correct the visual impairments. And I also mentioned this in my prediction video. Please know about astigmatism. Please, 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 please. How to identify it in a picture, how to treat it, and what causes it. Those 3 main things. Okay. Now, I actually want to pop over to the ear, and I'm just going to move this picture over a smidge. And then I'm going to zoom in on it. Okay. I'm not just going to do this one. I'm just going to list a whole bunch of things that I have emphasized to my own matrics about the ear, which is as follows. Please don't just know ossicles. Please also know the names of all the ossicle bones, which are from biggest to smallest, the hammer, the anvil, and the stirrup. Please know the name of those 3 bones. Don't just know ossicles. Please note that the hammer is the largest, then the anvil is the middle size, and the stirrup is the smallest. You don't need to know the Latin names. You can just use the everyday names. Please also know the function of the ossicles, that they are there for amplification. Please know that they are amplifying a mechanical movement and converting it into a pressure wave in the perilymph in the cochlea, which is what we can see here, right? So there is the oval window, which is exactly where the stirrup, that's the smallest bone. Let me make this bigger. Sorry, it's not very big. The smallest bone knocks against. Sound is going to go in through the perilymph, go around inside the cochlea, and it's going to stimulate, remember, the organ of Corti. That is the receptor you use for hearing, right? You're going to stimulate that. It's going to send an impulse via the vestibular nerve to your cerebrum, and you're going to interpret hearing. That's the 5-6 mark situation. But then we also have these other components. Let's not forget what the round window does. The round window is for excess reverberations or vibrations to leave the cochlea so you don't hear an echo. The next thing that we must learn, which is what's in this picture here, is these guys here, these semicircular canals. So let's go through all the possibilities. Semicircular canals, what are they for? They are there for speed and direction. Okay, so they're telling you what direction your head is going. Is it going up, down, left, right, forward, back? They love to ask this question when someone's running or someone is moving through space. And so again, what are you going to do? You are going to talk about how you are stimulating the cristae at the bottom and the base, which is this thing over here, and how it's being stimulated by the fluid moving and stimulating it. That then sends a message to the cerebellum. Remember, this is all about the cerebellum. And the cerebellum is going to send a message to your muscles of the body to maintain your posture and your balance. It does both. Remember the utricle and saccule, which sits over here. You've got one here and one there. They are responsible, the utricle and saccule, they are responsible for posture. So are you upright or are you lying down? That's why you have 2, because one is at a right angle to the other to tell you if you're lying down or upright. And yeah, basically, you also need to know the name of that receptor, which is the maculae. This one up here is the cristae. Must know those, is really important. And again, they can't ask that many questions, difficult questions on the ear. They can literally just ask you something about hearing, like balance or hearing itself. And then everything else is going to be like, label the diagram. What is the function of this? Why are the ossicles arranged from biggest to smallest? Amplification. What is the purpose of the round window? Gets rid of excess sound waves. What part of the brain does the cochlea communicate with? The cerebrum. So there's a lot of like content, knowledge, recall. It's not as much as you would think when it comes to explaining the ear, everybody. Okay. Nathan wants to know, why are there semicircular canals at right angles? They're at right angles so that you have one for every movement of plane of movement. So you've got to have one going up and down. You have to go one left and right and one forward and backward. That's how we move. We move in a 3-dimensional space, everybody. Okay. Right. And I think I'm going to run out of battery soon, everybody. So I just want to make that very clear. So I'm going to call this live very soon, but I want to just do a couple more questions in the background for you. Please put them in the chat now. About anything you want me to quickly answer or revise for you, please put that in the chat. So somebody wants to know, Feroza wants to know, do we have to draw any structures tomorrow? Please be prepared to draw a graph. Please be prepared to draw a pie chart, okay? I always threaten this one, and people think that I'm like being funny about it every year, but please be prepared for a pie chart, a graph. If they are going to make you draw anything tomorrow, it will never be something complicated. It'll be like a sperm cell, an egg cell, or like what the root or the shoot looks like after geo and phototropism have taken place. They're not going to ask you to draw like the whole ear. They're not going to ask you to draw the whole eye. They might ask you in a very unique circumstance to maybe draw I don't think they can't make you draw anything else. Egg cell, sperm cell, photo and geotropism results like of that. It's about it, guys. And graphs, obviously graphs. You've got to be prepared for a histogram, a bar graph, a line graph, and of course a pie chart. If my screen is blurry, it's actually not my screen, everybody. It is actually your screen. I'm streaming at a pretty high quality, so it just means that you need to change your settings in order to be able to see everything. I think I did make a mistake. Did I make a mistake? I'm trying to read what's here. Ma'am, can you do long-sightedness and short-sightedness type of lens to correct them? Okay, so I actually did that already. I spoke about it. I have myopia, which, by the way, everybody, you're supposed to know what myopia is. I don't have to call it short-sightedness. You need to know both. So I have short-sightedness. What corrects short-sightedness? A convex lens, convex. If I am long-sighted, which means I can see far away, or I have hypermetropia, I have a wait, hold on, rewind. Sorry, I have myopia. Myopia means I have a concave lens. That's the lens in my glasses. If I want to see, if I have far vision, which means I have long-sightedness, I have a convex lens in my glasses. Yes, that's why sometimes, if you've noticed, some people have that extra like window in their glasses, and it's like it bulges out, and that's because it is convex. Make sure I say that. No, please don't say that the round window releases pressure, because it doesn't. Tallulah, I'm going back to my picture now to see what you're referring to. that I swapped the lenses. Do you mean what you use to fix the lenses, or what the lenses are doing? You can reply to me. I'm not going to go off just yet. Somebody asked you to use a pen or pencil for graphs. You always use a pencil for drawing and a pen for labeling. I do have a pie chart video. I do actually. If you want to see a question with a pie chart, you can go into my question playlist on YouTube, the matric question playlist, and you'll see, if you look at the thumbnails, there is an actual picture of a pie chart. And I can't remember what the topic was. I don't know if it was for eyes or something like that, but you'll see it. You'll see the pie chart. I'll be in it. And then there's like a pie chart next to me as one of the graphics. That's a past paper question with a pie chart in it. So you can more than welcome to go and watch that. And I'll show you how to plot it and how to calculate everything. Calculations, that's a good one. You must be able to calculate percentage increase and percentage decrease, everybody, which is a very simple formula. It is initial minus final over initial multiplied by 100. So I'll say it again. Initial minus final divided by initial times by 100. And that will give you your percentage increase or decrease. It'll be a positive number or a negative number, depending if it's, increasing or decreasing. And by the way, if you are a member, everybody, it does also mean that you have access to my study guide. I'm just seeing, I do see Tallulah has a good little point for me that I'm just clarifying for myself, because I'm just going back in what I said to just make sure that if I do need to fix an explanation, I will do so now. I'm just going to go back and see. That's why if it looks like I'm looking all over the screen, is because I'm just going back in time to see what I had said about the eye, because I don't want to make a mistake as people watch this. And obviously, if I go too quickly, it's possible, and I don't want to do that. We spoke about accommodation, and we did the lenses. When you have myopia or short-sightedness, your eye is too long, the cornea is too flat, eye is too short, too far away. Oh, I think I know what you're talking about. You're talking about my accommodation explanation. We are on the same page. Don't you stress. So if I'm looking for near vision, near your ciliary muscles, they contract, suspensory muscles slacken. Oh, I'm with you, Tallulah. Okay, I'll share my screen quickly so everyone knows what we're talking about. So let me just open that up. Before I go, everybody, I want to clarify this. I made this mistake earlier, and it is an important one for me to fix before we go. These two are what I want to swap around. So this is close vision, and this is far vision. There we go. So it's about the shape of the lens, okay? And so it's all about the shape. The most important thing I want to teach you today, if